Hello, everyone. Welcome back after this coffee break. I'm chairing this session, and we are starting with Sam Riesenfeld from the Broad, and she will give a talk on using topic models to analyze the transcriptional spectrum of innate lymphoid cells.
Okay, hi, thanks very much. I'm happy to be here today to talk about some work that I've been doing in the last couple of years using single-cell RNA sequencing to understand a particular kind of rare immune cell that somebody already brought up once at least. So how does this, there we go. Which is on, this one is on, great. So tissue-resident immune cells control the body's immune response. And I've been studying this specific type, innate lymphoid cells, that respond very rapidly and early to immune stimuli, especially to signals from epithelial cells. And they can promote highly inflammatory responses. This sounds a little funny. Or tissue repair programs to help recover from damage. And we don't really understand what drives one response versus another. But we know that chronic imbalances in those kind of responses can lead to chronic pathologies and inflammation. So I've been using single-cell RNA sequencing to try to understand how these different responses arise, especially through signals from other cell types. So just to give you an overview of these cells, innate lymphoid cells are categorized kind of like, can you hear me okay? Sorry, I hear myself off and on. Great, I won't listen to myself. Okay, so they're categorized similarly to T helper cells as type 1, 2, and 3, typically, because they express the same sets of cytokines and transcription factors, and they contribute to similar kinds of immunity. I've been mostly focusing on ILC2s, which contribute to immunity against helminths, but also allergic response. And when those responses go awry, then we get diseases that are associated with the tissues where these cells reside. So I'm going to mostly focus this talk on some very recent work on a model of psoriasis in the skin, but I'll just introduce some of the ideas that we've come upon in the lung and also in the gut. That's like maybe a year and a half old, slightly older initially. So you can imagine they sit in mucosal and barrier tissue surfaces. So here I'm showing something like the lung or the gut. And the innate lymphoid cells, type 2, are the ones that are predominant in the lung. That's the ones I focused on. They respond to signals of tissue damage caused by worms making holes in epithelial tissue, and also to signals that we don't really understand their root cause, like allergens. And those alarmins, like IL-25 and IL-33, then drive different kinds of responses. So for example, you can get a tissue repair response that helps the tissue recovery and causes proliferation. You can also get very good inflammatory response, which helps clear the infections. And one of the things that we found is that signaling from a totally kind of, at least before the last couple of years, unconsidered kind of source is through neurons. So neuropeptide signaling from neurons can totally modulate this inflammatory response. It can really amplify it, as we showed it in an allergic context, but we've also seen examples where it can dampen the response and really shift it. And actually, what's interesting also is that this response involves both proliferation of local ILCs that are sitting there already ready to respond, and there's also been evidence that they get recruited sometimes, so that wasn't known before. They were thought to just be kind of only local. But some recent work showed that, for example, this highly pro-inflammatory subset in the lung actually gets recruited from the gut. So there's different mechanisms at play here. In this talk, I'm going to now talk mainly about how they drive skin inflammation in a model of psoriasis. And there we'll see that they actually I really liked the first talk, the box talk, because it introduced this idea that heterogeneity can be kind of a natural part of the cell state. And so we'll see that there's kind of a transcriptional landscape or spectrum that gets shifted by inflammatory stimulus, and that contributes to this inflammatory response. Okay, so just a model of how Psoriasis Works, very simple. So we know there's some trigger that causes keratinocytes, the skin epithelial cells, to signal to dendritic cells, which are kind of immune cell, and they use IL-23 signaling to communicate with T cells, both local T cells and recruited circulating T cells. And those T cells produce type 3 cytokines like IL-17 and 22, and that causes the keratinocytes to proliferate, to thicken, so you get these kind of thickened lesions on the skin. And then there's kind of a feed-forward cycle where you get signaling from the keratinocytes that causes that cycle to continue. And it's an extremely, extremely common immune condition. So it turns out that ILCs also play a role in this. That was learned in the last few years. They are also responding to IL-23 signaling. They're also producing 17 and 22 in response to that, as far as we understand. And they were seen to be in lesions, like in human lesions, you see an increase of them. So I just want to mention that this model, in the later models that we use in the mice, we actually treat with IL-23 because it's known that if you block 23 or 17 signaling in humans, you pretty much eliminate this disease. So it's clear that that's like a really important part of a really well understood part of the model. But what wasn't understood and still isn't totally understood is where the ILC3s are coming from. Because at baseline, basically you only see cells that look maybe ILC2-ish. We see kind of a variety. And we don't know if they're coming because they're getting recruited, like we saw in some cases in the lung, or if they expand from a population that's small, that hadn't been detected before. There's also evidence shown that there's precursor ILCs that was only seen in human, but they could be differentiating from those, or they could result from plasticity from other ILC types. And there's been each of these mechanisms has been observed in different settings. So single cell is a pretty useful tool in this kind of setting. So I showed you these really nice categories of ILC 1, 2s, and 3s, but really they have been seen in specific kind of contexts under specific triggers to get pushed into a different state. So we know that they can move between 2 and 3 or 2 and 1 in different tissue contexts and inflammatory contexts. And then I said there's these ILC zeros or precursor cells that were seen in humans. So we were really set out to just figure out what was going on here. And so I'm mainly computational, but I worked very closely with a postdoc in Richard Flavel's lab at Yale, who was doing these experiments where he injected subcutaneously in the ear IL-23 for 4 or 5 days in a row, and every day measured the increase in the thickness of the ear, and then at the end took a sample of the tissue. We did a bunch of different downstream experiments, including single-cell RNA-seq. So the first thing is just to establish this model is working, and we do see this increase in ear thickness due to the 23, and we're pretty sure that ILCs are contributing a part of this, so it's very complicated. I mean, if anyone's done immunology, you know it's very complicated. All these different cells are working together, including a different kind of T cell that's local. But basically here I'm just showing you that this is kind of the baseline that you get if in a mouse that's got no T cells and no ILCs. It's a RAG gamma common chain deficient mouse. And here's a RAG deficient mouse with just no T cells, but it has ILC. So you see this is kind of the baseline from just irritation, and then you do get inflammation, and then from there, when you have both T cells and ILCs, you get this amplified response in the wild type. So we wanted to understand this part that's contributed through the ILCs, and we also tried immediately this experiment that was used to show the recruitment to the lung from the gut. So you treat with this treatment called fingolimod or FTY720, which is supposed to block trafficking of T cells and to some extent ILCs. And in that setting, it prevented the ILC2s from getting to the lung. In our setting, it seemed to make no difference at all. So we assume either this isn't the right experiment for us to be doing, or there's enough local effect of ILCs. Maybe you don't need recruitment. OK, so then that leaves the possibility that they're either getting expanded or that perhaps the population that's there is actually poised to become this other kind of type of ILC. And so we see exactly that evidence in the ATAC-seq data. We see that it's true that at the start sites, and the untreated wild-type mice, you basically you only see open chromatin at the transcription start sites for type 2 cytokines like IL-5 and IL-13, which are characteristic of ILC2s. You don't see it at the start sites for type 3 cytokines like IL-17 and IL-22. But what you do see is that if you look at elements that were identified in ChIP-seq studies for Th17 cells, which are express the same kind of type 3 cytokines, you see that there are enhancer elements that look like they have accessible chromatin in the attack-seq. So that gave us this idea that maybe they're kind of poised to become ILC3-like, which is a different model, actually, from how T cells get differentiated. So we wanted to look then at the single-cell data more closely. This is an overview of it. And so I looked at ILC data before. It almost always looks like this, honestly. If anyone's done an ILC study, you'll almost always see like a giant cloud. It just doesn't tend to separate into really specific substates. And of course, so here I'm showing you the untreated and induced. So this is untreated, this is induced. Here's wild type mice, and this is RAG deficient mice. So you see almost all the cells fall into this kind of big cloud. There are some specific clusters. The main one that we're interested in is this cluster here, which is unique to the induced condition. So there's almost no cells from the untreated condition there. And if you, this other cluster I can talk about a little bit later, but it's due to cell cycling. So it's just pulling cells from all of the different conditions because they're cycling. And here, this is one that we kind of are going to ignore for now because it's specific to the RAG deficient mouse, and we're more interested in the natural physiology, which is in the wild type mouse. We just were using this to check that it was reproduced when you only had ILCs, but some things do change without T cells. So you can cluster this data, but the problem is that you basically end up kind of subdividing patterns. If we already know about some of the key genes in these immune cells, you end up seeing these clusters kind of subdividing them in weird ways, and it doesn't feel like it's like characterizing the cloud very well. And of course, that could be due to noise, and I think there's definitely a technical component of this, but it does seem also structured in some way. So so for just the other thing it doesn't capture, clustering doesn't capture, is the fact that you see signal of important genes. Like this is considered a type 2 gene, but it's crossing the cloud and into this kind of type 3 cluster. And that's a hard kind of thing to pick up with differential expression analysis that just subdivides and tries to compare clusters. So we wanted to try to figure out if there was like a more natural way of characterizing this. And I just wanted to say that there's not really any good reason why we tend to do this kind of subdivide in differential expression, except that some really early great studies did that, laid out kind of this pipeline. And they had data that looked like this. This was done in our lab on retinal bipolar cells. There were tons of different cell types. They had really clear morphological differences. They segregated really well. And it made a lot of sense to do that. And it doesn't make sense when you're zeroing in on a more specific cell type and you're looking at different responses, or even just at baseline. I mean, some of this is just variation that you see at steady state baseline. And so if we want to characterize what that variation looks like. So I just want to talk for 1 second about how you can how I at least imagine how this kind of variation might arise. You could imagine that suppose you've got like 3 seminal programs that are going on, like gene programs that are going on. You might have a red program and a blue program and a yellow program. And you don't have to ever, so we know that we're probably not going to get a cell that expresses just one of these programs, right? You're not going to get these corners most likely. But if you just know, if you think about them being there, it's still really a useful basis for explaining this whole mixture of all these different states that you could get, right? Because they're all basically a mix of these 3 programs. So you get this like infinite space that's really clearly, that can be really clearly described by saying what proportion of red, blue, or yellow each cell has. So it's natural, right? We use those kind of descriptions all the time, but we haven't used them that much in single cell. Another place that this has been used, this kind of analysis is common in a few different areas, but one place that was a natural analogy for us was in document analysis. So you can imagine there's this analogy between text analysis where a document corresponds to a cell, and then the word in a document corresponds to just a single gene transcript appearing in the cell. And so when you're doing certain kinds of document analysis, you might be trying to just annotate all of the themes that are important in a body, that like an online corpus of documents, and you don't want to read each one. You just want to kind of know from the words that are in each document. And that might be something similar. You're trying to figure out what are the kind of general cell states that are covered by the cells in this space without necessarily looking at every gene in every cell. Or every transcript in every cell. So you're trying to characterize the processes or the activities or the cell states, depending on what word you feel comfortable using. So this was really popularized in text analysis. Like I said, I mean, it's just a probabilistic model. This version is just a probabilistic model over non-negative matrix factorization. It has pros and cons, which I can talk about for a minute at the end. So here you can just think about it. Sometimes it's easier to think about the text first. You can think about the generative process is basically each word is generated by a specific topic, and how likely that word appears depends on how what its weight is in a specific topic, and also what that topic's weight is in the cell. And so you're trying to infer both jointly the topics, which are just distributions over words, and the topic weights in the cells jointly, so that they explain the data well. Here I just want to say, so here these, basically you'd be reading along and you'd say, oh, I see, I keep reading documents where I see DNA and gene and maybe RNA or whatever, something all together. So maybe there's like that's a topic, because I keep seeing those words co-occurring together. Similarly, you might have these other ones, and words can appear in the same. They can appear in more than one topic, and just like a cell. So it's a kind of a way of soft clustering the cells. So what does this look like in single cell? You might have, so in single cell, you imagine each topic is a distribution over genes, and the number of transcripts that appear, the count, is related to the weight of the gene in the topic, or the proportion of the gene in the topic. And then you can describe each cell as a set of proportions describing how much each topic contributes to the transcriptional profile of that cell. So this cell has a lot of blue and a little bit of orange. This one has a lot of orange and just a little bit of blue, and so on. You can get different mixes, and that helps explain the transcriptional profiles that you're seeing in each cell. And you're trying to infer these together, so nobody tells you either of these. You just you optimize them both so that together they explain the data that you see well. Oh yeah, one other thing about this model is that this is just a simple kind of vanilla version of this model. It's called latent Dirichlet allocation. I don't know if I mentioned that when it came up. One thing that's interesting about using an explicit probabilistic model is that we can think about how we might tweak the probability distributions so that we can maybe try to explicitly model dependencies and things like that, which I haven't done at all in this case. I just was kind of experimenting with trying it out. But that's a place to think about going. So what does it look like when you actually apply this? We found it really helpful. So here are a couple of different topics I got. I say, okay, choosing the number of topics is a trick, just like choosing the number of clusters is a trick. Whatever, all we do is kind of art. It's not really scientific, right? But this is but here we you can run it with a few different numbers and see how stable it is and so on. But the point is that I want to show you is that you get these kind of different topics. So this topic basically captures what you would get by doing cluster-based analysis. So you can get topics like that are almost like differential expression. Then you also get topics like this one that it's mostly in this cloud, but it gets this graded expression across. And I named this repressive mainly because I looked at the genes that were kind of very featured in that topic and were specific and upweighted in it, and saw that they were also associated with a lot of T-cell repression programs and Th17 repression programs, and I thought, okay, maybe that's maybe that's actually up. And so it was also up in the untreated condition, so I thought maybe it's like actively repressing this ILC3 program that's sort of poised behind the scenes. This was the reason for the name, but I wasn't given that. And the same thing here, this had lots of type 2 genes that I knew, so I named this type 2 and named this mixed type 3. This one particular caught our attention because it managed to bridge these two clusters, capturing that program. So do I have any sense of what time it is? Is there a clock or anything? What does that mean? Huh. Okay. How can I tell that from that? Oh, my God.
It will
Okay. All right. Great. So, yeah, that's fine. I can do it. I can divide by 5 or 6 or whatever that is. I can do that. All right. So just as a sanity check, like this analysis says, and you could get this lots of ways, of course, but this analysis suggests right away that you have these co-producers. You have cells that are producing both what are usually thought of as type 2 cytokines with type 3 cytokines. And when we, checked using FACS staining, we did actually see that. So we were sort of heartened that this wasn't just a bunch of doublets or something like that. You see both 22 and 17, these type 3, you see 13, and then you also see these double producers. So then trying to figure out what you're going to do with this analysis is, I think it's also, again, kind of a matter of exploration and art right now. This is just an example of how you might try to use it. I knew that I was particularly interested in understanding how these kind of topics, how these programs were related to each other, both within the steady state and moving from the steady state to the inflammatory state. So it was actually, there were, a lot of the time when you analyze immune activation in single cell, you actually see what most people would consider technical kind of programs that are very strongly correlated with the actual activation because the cells get a lot bigger. And so you get RNA, tons of RNA programs, and that's not necessarily wrong, it's just not necessarily interesting. So I basically focused on the programs that had immune-related genes that I thought might have something to do with what I was interested in. So I took cells and genes that were upweighted in those programs, and I excluded cells and genes that were either upweighted in that RAG-specific program, or that there was one that looked like it had like really high mitochondrial kind of content in the program. So you can also use it to kind of clean up your data because usually that's associated with poor cell quality or unhappy cells. All right, so I still grabbed a pretty good distribution of cells, and then I used those to do a diffusion analysis because I just wanted to understand how in high-dimensional space these different states were relating to each other. So what you get out of that, the first 3 components show you the main features, as far as I could tell. So you get basically most of the cells from all of the cells from the untreated state lie in this kind of triangular plane here in diffusion component 1 and 2, and then you get kind of another diffusion component coming off here, which seems to be in the direction of perturbation. So if you focus just on this part, there are actually a few different topics that are relevant for the cells here, but each of the sort of corners is like extremely upweighted in one of these topics that was kind of upweighted in the untreated cells. So it's like they can kind of explain, like every cell in that untreated group is a mix of these topics to some extent. And then I wanted to focus on this transition. So I focused in on 3 topics that really seem to capture the middle and ends of this transition. And looked just at a diffusion map of those cells and genes, which went from this repressive topic through this type 2 topic into this type 3. There's probably other possible trajectories, but this suggested this, very clearly, this trajectory that if you want to get from, say, a repressive topic, which is upweighted in the untreated cells, to this type 3 topic that's really strongly upweighted in the treated cells, you have to go through this type 2, which is the kind of result I was wondering about. Like, is this an actual activation pathway or not? Now, of course, this is computational, and it doesn't prove anything, but we were actually able to validate this part of the predictions by using a fate tracking mouse. So IL-5 is this type 2 cytokine, and it's really, even though this topic 13 is expressed all the way along here, IL-5 is really, really specific to these cells up here. So we were able to do a fate tracking mouse where you can track whether a cell has ever expressed IL-5 with this YFP. If it's currently expressing IL-5, you'll see it with ROSA, and then and then we also crossed it with a 17 and 22 reporters. So you could basically see active 17 or 22 translation. And so because these are like these are small numbers of cells and even smaller numbers if you're looking at the specific pathway, we did a re-challenge to help boost the number of cells. And you see that you do get increased numbers of 17 and 22 according to the reporter. And you get maybe a small increase in 5. But what was interesting to us is you definitely get this boost in cells that once, as part of their history, expressed IL-5, turn that off, don't express it anymore, and turn on 17 and 22. So that is, in fact, a pathway that does seem to be a valid trajectory for activation. It's hard because this reporter isn't that faithful, and so we probably lose a lot of cells that might actually be going through that pathway but just don't get stained as being positive. One thing is that we know that IL-5 goes off because we see almost no double producers. So if you just look at a snapshot, you would never think that cells actually go from one of those states to the other because you basically see no co-producers, but for 13 you do. So it looks like those cytokines, which are usually considered to be hand-in-hand for type 2 immune cells, are actually differentially regulated in this process. OK, so I think now it's like going the other way. No, but it was higher 1 second ago. I can't follow that thing. OK, I'm sorry. Yeah, I think I'm done, right? OK, good. I'm at the end, so just making sure I'm not ending half an hour early. Like that thing makes me feel like it's the middle of my time. OK, I can give my next talk. No, OK. So just to summarize, this are skin-resident ILCs. They look like they're sufficient without T cells to drive this kind of skin inflammation. And we showed that even at steady state, there's this dense continuum of transcriptional states. I think this is kind of, by today's standards, a small number of cells and a focused analysis, but I think it's really useful to think about how we can get these kinds of analyses right. Like, how can we characterize variation, even steady state variation, in transcriptional space and model it, and then try to extend that to larger scale analyses where you have multiple layers, and you also have cell types. And then we were able to validate this dynamic activation in vivo. So one note is that, like I said, I think it's important to try to experiment with the probabilistic model. You could look for you can try to get cell type hierarchy or cell state hierarchies or correlations. For example, probably some programs are much more likely to be together or anti-correlated, and this doesn't capture any of that explicitly in the model, so you could try to incorporate that. It just makes it a little bit harder to infer the model. But there's some newer ways to think about doing that. The other thing that I'm interested in is, I guess I don't really say it there, but I think it's interesting to think about drilling down to what we can learn about genes in continuous space representations at a really low level, because we now have so many different single cell samples. So I'm also kind of going in that direction. And last, just want to thank my collaborators. This was a great collaboration with the wet lab, Richard Flavel's lab. Piotr did all the experiments. Monica used to be in Aviv's lab and did the genomics. Richard Loxley helped us a ton recently to get some more data with the IL-13 fate reporter. There's lots of stuff I didn't show you. All right, thanks.
Thank you. Maybe first question here.
Hi. Did you binarize the count matrix for the topic analysis?
No, I didn't. Are you going to ask me about how some genes are much more highly expressed than other genes or something like that? I didn't binarize the count matrix, and you don't need to get good results this way. You do need to for certain kinds of co-occurrence types of analyses, but you don't need to for this, actually. It works pretty well. There are ways of trying to, in some sense, normalize either the variance without normalizing the mean, or log normalize, or somehow put the different scales of gene expression a little bit more on the same level. Honestly, it doesn't radically change the answers that I get. Somehow, I think the correlations are strong enough that they kind of get teased out, but that might be necessary for some settings. There was other questions, I think, back there.
Thank you, very interesting talk. When you work with probabilistic models,
yeah.
Did you try to use any prior information, maybe by cell types, I mean cell markers, to put more biological meaning in these topics?
Yeah. No, I didn't, and I didn't on purpose, because our knowledge about these comes a lot from T cells, and it's often like I just wanted to go fresh without. But it's definitely, I think it could be a reasonable idea, especially if we start doing some kind of hierarchical type model, or if we're learning from more different cell types, then you might actually want to have that in there. This was just also, it was a very limited cell type So I wanted to see what de novo came out of it in terms of the cell states. You know, a lot of what's out there, it's like we keep using it to annotate what we have, and then we don't ever learn new things, and some of it isn't that robust, really, or it doesn't generalize that well. So I try to also do from scratch sometimes. I think there was one more, yeah.
All right. So you looked in the skin, which is more Th2 prone. What do you expect in the gut setting where you have more Th17, ILC3 setting? Do you see this ILC2 signature there as well?
Yeah, you do see ILC2s there. So I'm not an expert in the gut, but just as an example, that neuronal sort of response that I showed is actually 2 other labs found it at the same time. That was with a Vijay Kuchar, so the different lab at Brigham, but our work and 2 other labs were all found this kind of neuronal ILC2 signaling and the amplification of the response specifically to allergens or helminths, and they were looking in the gut and we were looking in the lung. So it's definitely active. You're right that you see more ILC3 at steady state, but they're still present and they can expand. Sorry.
Topic models are like, at least in simplest form, matrix factorization models, right? Right. So
just a generative model on top of matrix factorization.
So you could do like an NMF, but there were these early approaches. We had one, and then I think Ollie was following up with his SCLVM, you know, where you put some informed things in there. Yeah. How does that compare? Because in that case, you know, we made a big fuss about, you know, including the noise model, which, you know, I guess you could also do at some point. Right. It didn't really matter much in your situation, you said, right?
No, yeah, I think.
Did you see cell cycle, for example, one of those topics?
Oh yeah. Yeah. You see cell cycle.
Focused on one?
What?
Focused just on one topic, so on one component of the matrix?
I think so, yeah. In this case, yes. Sometimes cell cycle for me is really, because the response can induce proliferation, you sometimes see it like all mixed up. Like it won't be combined because it's such a strong program, so it always pulls out separately, but it's actually aligned in terms of the cells that are expressing it very well with the other topics. But here, it's actually, there's not a lot of proliferation in this particular response. So it's nice because for once it separates. Yeah, you see it basically, it's basically like a cluster analysis for that. And you could just like throw that out and take.
I think slalom is what they actually ended up calling it.
Oh, sorry, I can't go back. But just as an example, like the topics that I used, one of the things that's nice about it is it actually splits up that cell, just like you would do with NMF, it splits up that cell cycle cluster. And so I could grab pieces of it that actually belong to other topics and just ignore the cell cycling because I didn't want to pay attention to that. But it could. But if there were some that were expressing type 2 or type 3 or whatever, they were all getting pulled in there, and so you can split it up this way.
I mean, and there should be a negative binomial NMF around, right? Which would sort of then adapt to the noise model, I guess.
Yeah, I mean, there's been all this recent work that multinomial is the right thing, and this is a multinomial, so maybe this is the right thing. I don't know. Okay.
Yeah, I think that was the last question. Thank you, Sam.
Okay, thanks a lot.